Hello everyone, my name is AJ Keefe. I'm going to be presenting on this excellent paper titled Aging Triggers Cytoplasmic Depletion and Nuclear Translocation of the E3 ligase Mahogunin, a function for ubiquitin in neuronal survival. I want to give special thanks to the authors down here and the Journal of Molecular Cell. During this presentation, I want to recommend that anyone watching pauses the video at each new slide and reads through the material and digests the figures that I have included. I think that'll help you grasp the material, and I hope you enjoy this presentation. The authors of this paper are particularly interested in this E3 ubiquitin ligase, MGRN1, because its deletion in mice causes an age-dependent neurodegenerative phenotype. Additionally, MGRN1 has been shown to protect against oxidative and endoplasmic reticulum stress, protect against prions and polyglutamine aggregates. How exactly MGRN1 promotes neuronal survival, however, is not understood. Thus, the current paper aims to characterize MGRN1 and discover exactly how this protein is protecting neurons. And this is what they found. First, they observed an age-dependent decrease in overall MGRN1 expression. Additionally, MGRN1 that remained in aged mice was almost exclusively located in the nucleus, distinctly different from the cytosolic localization observed in young mice. Since MGRN1 is a ubiquitin ligase, the authors decided to see if the proteasome activity was perhaps orchestrating the relocalization of MGRN1 from the cytosol to the nucleus. Indeed, they discovered that proteasome inhibition was sufficient to cause MGRN1 translocation to the nucleus, and furthermore, this relocalization of MGRN1 was contingent on its monoubiquitination. The authors noted that two proteasomal-bound deubiquitinases, UCHL5 and USP14, both were causing the deubiquitination of MGRN1 and thus were promoting its localization in the cytosol. Next, the researchers sought to characterize the activity of the internuclear MGRN1 that was observed almost exclusively in the aged mice. Using next-generation RNA sequencing and ChIP-seq sequencing, they found MGRN1 to be intricately involved in mounting the transcriptional response to proteasome dysfunction. Among the many genes upregulated by MGRN1 was ATF3, a stress-induced transcription factor that upregulates pro-survival genes to counter the proteasomal stress. And indeed, MGRN1 was critical for the expression of ATF3. Furthermore, MGRN1, its upregulation in vivo through lentiviral transduction, enhanced memory and cognition in aged mice in various memory tasks. These results collectively suggest that MGRN1 is critical for the transcriptional response to proteasomal dysfunction, and its age-related decline may underscore various neurodegenerative disorders. The researchers began by examining the subcellular localization and total expression of MGRN1 in young and old mice. They found that in the post-nuclear supernatant, the PNS, or the cytosolic fraction, MGRN1 was enriched in young mice, but was markedly missing in aged mice. So although 24-month-old mice have comparable total levels of MGRN1 shown here, the post-nuclear supernatant of their cell lysates was missing MGRN1. This suggests that MGRN1 was primarily found in the nucleus of aged mice. Immunofluorescence assays confirmed this by peering directly into the cells and observing a primarily nuclear localization of MGRN1 by its co-localization with the nuclear stain DAPI. So you can see that the MGRN1 expression in 5-month-old mice is highly diffuse, while in 24-month-old mice, the MGRN1 staining in green is highly co-localized with DAPI. So these experiments suggest that total expression of MGRN1 in old mice is roughly 70% that of young mice, But more interestingly, the cytoplasm of aged neurons contains only roughly 20% of the MGRN1 found in young neurons. So we see a massive change in the location of MGRN1 as these mice are aging. Having shown a profound age-dependent relocalization of MGRN1, the authors then tried to determine potential triggers, and they began by looking at the proteasomal dysfunction, since MGRN1 is itself a ubiquitin ligase, and because the neurodegenerative disease that's caused by knocking out MGRN1 is associated with proteasomal dysfunction. So it's likely interconnected in some way. So a well-known inhibitor of the proteasome is this MG132. We're going to be talking about that a lot. So MG132, in this graphic right here, was administered to these cells, and then what they observed was that in the presence of MG132, MGRN1 localizes to the nucleus. The nucleus is in white. We see vast upregulation of MGRN1 in the nucleus when MGRN1 is applied. So when we're inhibiting the proteasome, we're seeing translocation to the nucleus as opposed to these control cells treated with saline. So next, they decided to look at the cytoplasmic and nuclear fractions so that you can see over time. So 28 hours after MG132 treatment, we see almost no cytoplasmic MGRN1 as indicated by this Western blot of the cytoplasmic fraction. And in graphical format, we also see a time-dependent decrease in the amount of MGRN1 in the cytoplasm following proteasome inhibition. In contrast, we see the exact opposite pattern in the nucleus. So as proteasome time increases, going from left to right, we see at 28 hours after proteasome inhibition, we see a ton of MGRN1 in the nuclear fraction. So in graphical format, again, we see increasing amounts of MGRN1 over time following inhibition of the proteasome. And then visually, we also see this in immunocytochemistry. We can see that MGRN1 localizes in the cytoplasm in control cells, and in the DAPI-stained nucleus, we don't see any MGRN1. In contrast, when you inhibit the proteasome using MG132, we see translocation and sequestering of MGRN1 in the nucleus. We see the nucleus is chocked full of MGRN1, as indicated by the colocalization. So proteasomal inhibition using MG132 was sufficient to induce nuclear translocation of MGRN1. How then does proteasome inhibition lead to MGRN1 translocation to the nucleus? To test this, the researchers decided to look at monoubiquitination, which is a common mechanism of tagging proteins for transport. On the right, we see the results of an immunoprecipitation of ubiquitin, so the immunoprecipitating ubiquitin, followed by the immunoblotting or the Western blotting of MGRN1. So this immunoblot reveals that co-immunoprecipitation of MGRN1 increases following treatment with MGRN1 for ubiquitin lysis. This means that MGRN1 is ubiquitinated following MG132 treatment. So let me reemphasize. They are immunoprecipitating ubiquitin, so they're pulling down ubiquitin, then they're staining for MGRN1. And they do this in two different conditions, a control condition with DMSO saline, and then they do it with cells treated with MG132. And we see cells treated with MG132 are pulled down more frequently than cells treated with a control. And this indicates that there's more ubiquitinated MGRN1 when the proteasome is inhibited. So the question is, could this ubiquitination be triggering nuclear import? To test this, they screened MGRN1 for ubiquitinated lysines using mass spectrometry, and then they mutated all of these lysines to arginines such that MGRN1 could no longer be ubiquitinated following MG132 treatment. And these MGRN1 proteins that lacked lysine could not translocate to the nucleus following MG132 treatment, suggesting that ubiquitination is critical for the process of translocation. To confirm this, the researchers generated an MGRN1 protein that was fused to a mutated ubiquitin protein that lacked lysines. So they have this monoubiquitinated MGRN1 protein that contains a mutated ubiquitin that cannot be polyubiquitinated. Then they transfected this protein, and they observed it translocating to the nucleus. So the presence of a single monoubiquitination on MGRN1 was capable of translocating to the nucleus. Additionally, the researchers found that inhibition of CRM1, it's a nuclear export protein, caused the nuclear accumulation of MGRN1. So this suggests that CRM1 is responsible for the export of MGRN1. So they're starting to uncover the pathway that's going on. And also this Western blot down here, what they did was they fused MGRN1 to a wild type ubiquitin that could be polyubiquitinated. And you see the amount of MGRN1 following proteasome inhibition. And they also have ubiquitin lacking lysines right here. So when the ubiquitin that was fused to MGRN1 was lacking lysines, there was slightly less ubiquitination. So right here we see the polyubiquitinated MGRN1 because it's a wild type ubiquitin that was fused onto it. Then we see the ubiquitin that lacks lysines that was fused onto MGRN1. And we see less pull down of MGRN1. So this indicates that MGRN1 is being polyubiquitinated in that the mutated lysine on the ubiquitin does indeed make sure that it's monoubiquitinated, and it does prevent polyubiquitination. So this is done just to confirm that these lysine mutated ubiquitin molecules that are fused onto MGRN1 are maintaining its monoubiquitination. Since proteasomal impairment caused the nuclear translocation, the authors wondered if proteasomal associated deubiquitinases played a role in MGRN1's dynamic ubiquitination process. To test this, they used a small molecule inhibitor, which is BAP15. We're going to be seeing that used across here. BAP15, which is an inhibitor of two proteasomal deubiquitinases, UCHL5 and USP14. So on the bottom left, we see the results of an immunoprecipitation of ubiquitin, followed by staining of MGRN1, and the presence or the absence of deubiquitination inhibition by AP15. So we can see that when these proteasomal deubiquitinases are inhibited, the amount of MGRN1 that is ubiquitinated increases to a level that is comparable to MG132. So we see, especially under higher exposure, it's kind of hard to see right here, but under high exposure, when this is the saline treated condition, so we're pulling down ubiquitin, then staining for MGRN1, so these bands indicate MGRN1. In the saline condition, we see almost no MGRN1 pull down, which makes sense because it's not ubiquitinated. And then MG132 treatment with the proteasome inhibitor causes vastly increased amounts of ubiquitinated MGRN1 that's been confirmed previously. And then an inhibitor of the deubiquitinasers, the UCHL5 and the USP14 deubiquitinases, that caused increased ubiquitination as well, suggesting that MGRN1 is deubiquitinated by these two ubiquitinases, and that inhibiting them causes increased ubiquitination of MGRN1. So inhibiting the deubiquitinases yielded a significantly increased amount of MGRN1 ubiquitination, and presumably this would be accompanied by increased translocation into the nucleus. So in other words, the UCHL5 and the USP14 deubiquitinates MGRN1 for either subsequent proteasomal degradation, because it needs to be deubiquitinated before it goes into the proteasome, or it's also possible that the MGRN1 is escaping right before it's completely deubiquitinated, when it's monoubiquitinated, and entering the nucleus. Either way, on the right, we see the result of UCHL5 knockdown using short hairpin RNA. So in this condition, they're using short hairpin or siRNA to knock down UCHL5. And when you do that, you see this increase in the band right here. And this is actually monoubiquitinated MGRN1. So when you inhibit the deubiquitinase, you get more monoubiquitinated MGRN1. So these results collectively suggest that the monoubiquitination of MGRN1 is increased following the knockdown or the inhibition of UCHL5. And so UCHL5 is the primary deubiquitinase that cleaves the ubiquitin from MGRN1. Now that the nuclear translocation following proteasome inhibition and monoubiquitination has been established, what importance does this process have? So does it actually matter that MGRN1 is entering the nucleus? So to examine the nuclear location of MGRN1, the authors conducted first an immunogold electron microscopy experiment, which is not shown here, but what they observed was a non-random localization of MGRN1 in primarily euchromatic areas, so areas of active transcription. Then a Western blot analysis of chromatin fractions revealed an increased amount of chromatin associated with MGRN1 in aged mice compared to young mice, normalized for histone 3. So the same amount of histone 3s. There's just less MGRN1 in young mice, lots more in older mice. Then lastly, they did immunofluorescence analysis of the nucleus labeled with DAPI, just to look at the nuclei and the euchromatic areas, which is these the non-blue areas, because DAPI stains the heterochromatin mainly. And they also used PSR, which is a marker of nuclear speckles where RNA is processed. And since chromatin regions surrounded by nuclear speckles, marked by this PSR, are known to be active regions of transcription, the co-localization of MGRN1 with PSR suggests that MGRN1 is accumulating in regions of transcriptional activity. So collectively, these results all suggest that MGRN1 is found in transcriptionally active regions of the nucleus. In order to determine if MGRN1's nuclear translocation resulted in changes to transcription, the researchers decided to conduct a shotgun mRNA sequencing experiment to analyze differential gene expression. So they were comparing the transcriptome of cells transfected with MGRN1 GFP and the proteasome inhibitor MG132. So this is the experimental group, and they were comparing that to control cells transfected with GFP and also treated with MG132. So the only difference between these two cell groups is the overexpression of MGRN1. So any changes in transcription could be attributed to the overexpression of MGRN1. And they discovered that cells transfected with MGRN1 had upregulated numerous genes that were primarily involved in ontologies related to ubiquitination and the reactive oxygen species response. So we see upregulation of all of these genes and many hundreds more probably, but this is just a subset. We see relative mRNA levels compared to the control condition. So right here, this would be, roughly 5 or 4 times increased expression of FOSL1. So we see upregulation of all these genes, and although not shown here, they also did a chip sequencing experiment towards RNA polymerase II. So they are immunoprecipitating RNA polymerase II, and then they're sequencing all the DNA that gets pulled down with it. And what they found was that the RNA polymerase occupancy of all of these promoters right here is upregulated. So not only is there more mRNA, but the occupancy of the RNA polymerase enzyme itself is increased at all of these genes. And even more fascinating, just to confirm this, they also did a ChIP-seq towards H3K4 methylations. So H3K4 is an activating methylation found at active gene promoters. and they found that H3K4 methylations were increased following treatment or following MGRN1 upregulation. So collectively, all these results put together suggest that MGRN1's translocation to the nucleus is not a passive process, but is a very active process that drives gene expression by reconfiguring chromatin. ATF3 is a major stress-induced transcription factor that was further investigated because it was upregulated in MGRN1 treated cells, and as we can see right here, these other two genes, FOSL1 right here and PCK1 right here, it just so turns out that these two genes that are highly upregulated have ATF3 binding sites in their promoter. We also see ATF3 is upregulated to a highly significant amount, I should note. So because these two highly upregulated genes, which contain ATF3 binding sites in their promoters, and because ATF3 was upregulated, they decided to look into ATF3 expression. So in the top panels, we see that MG132 treatment right here, and thus inhibition of the proteasome, induced expression of ATF3. So it's upregulated when the proteasome is inhibited. We see that MGRN1 expression alone, right here, is not really able to induce ATF3 expression, probably because it's in the cytoplasm. But then when MGRN1 is added with MG132, so when the proteasome is inhibited, when MGRN1 is upregulated, you see ATF3 expression is potentiated. So we see even more ATF3 expression than proteasome inhibition alone. We can see it in graphical format right here. So and down here, we're seeing the effects of short hairpin RNA. So we're inhibiting the expression of MGRN1. So we see when MGRN1 is inhibited by itself, it's not too interesting. So right here, we see this is basal ATF3 expression. This is what happens under normal conditions. This is what happens when MG132 is administered. So this right here is essentially the same experiment as this right here. We're seeing increased ATF3 expression when MG132 is administered. What's interesting, though, at this point, is when you knock down MGRN1 while simultaneously administering MG132. So MG132 and proteasome inhibition is occurring right here. And we don't see any ATF3 expression. What this means is that the presence of short hairpin RNA towards MGRN1, so the knockdown of MGRN1, is capable of completely preventing the ATF3 response to proteasome inhibition, which is really quite amazing. I mean, this suggests that MGRN1 is required for ATF3 expression, because when you knock down MGRN1, the proteasome inhibitor MG132 cannot mount the expression of ATF3. So this suggests that an age-associated decline in MGRN1 might cause a decline in ATF3 expression. And since ATF is a pro-survival transcription factor that responds to proteasome inhibition, this might be a mechanism by which MGRN1 dysfunction is causing the loss of neurons. Having established that MGRN1 responds to proteasomal dysfunction by upregulating ATF3, among many other genes, the authors wondered if this, at a macroscopic level, at a macroscopic scale, would produce measurable behavioral changes in the living organism. So to test this, they transduced MGRN1 using a lentivirus into the dentate gyrus of the hippocampus in mice. So this is basically the learning center of mice, and they basically injected MGRN1 using a virus into the hippocampus, and then 3 months later, these aged mice were tested in 2 behavioral tasks aimed at memory and general cognitive function. Mice transduced and overexpressing MGRN1 has significantly better memory, as assessed by these tests, and significantly better general cognition, as assessed here. So these results represent the kind of coup de grace of their experiments, suggesting that MGRN1 mediated transcription in response to proteasomal dysfunction is indeed a very physiologically relevant process. And you can see this beautiful image here shows us the immunocytochemistry and the expression of the MGRN1 GFP transcript that was transduced into the hippocampus. So what has this research revealed, and what does it actually mean for the future of research? So remember that MGRN1 is an E3 ubiquitin ligase, and that previous research has identified it as being neuroprotective in various models of neurodegeneration. And additionally, remember that deletion of MGRN1 causes an age-dependent neurodegenerative phenotype. So we know that MGRN1 plays a critical role in the survival of neurons. We know that right off the bat. But many other genes also play a critical role in the survival of neurons. But what is perhaps unique and interesting about MGRN1 is that this normally cytoplasmic protein responds to proteasomal stress by translocating to the nucleus. And there it mounts the subsequent pro-survival transcriptional response, mainly through ATF3. Thus, MGRN1 may be the primary signaling route by which the proteasomal stress is being translated into compensatory gene expression for survival. So that's, all of that's pretty amazing. But in the end, we should always be left with more questions than answers. And here are a few of my questions that I was left with at the end of this paper. So if MGRN1 translocation to the nucleus is a response to proteasomal dysfunction, do disorders of the proteasome show robust MGRN1 relocalization and gene expression like we might expect? So throughout this entire paper, we've been discussing proteasomal dysfunction only in the context of MG132 inhibition. And MG132 does not inhibit the proteasome in disease. So what might happen if we take a model of disease which has been shown unequivocally to inhibit the proteasome, such as the C9orf72 mutation that causes ALS? That mutation has been shown to inhibit the proteasome. So does that mutation also cause MGRN1 translocation, as we might expect? And how does ATF3 or other components of the MGRN1 transcriptional response promote cell survival? Can the critical proteins derived from MGRN1's transcriptional response be teased out from the background static? So can we actually find out which proteins are promoting survival? And is MGRN1's ubiquitinase activity required for its transcriptional response? So I was actually a little surprised they didn't investigate this in the current paper, since they did generate a ubiquitinase deficient MGRN1 plasmid. So I wonder if its ubiquitination activity is a mechanism by which it primes or promotes gene expression, which I imagine it would be. What proteins associate with MGRN1 and perhaps modulate the process of translocation and gene expression? I think an unbiased screen of MGRN1's interactome could be very insightful. And finally, is nuclear translocation of MGRN1 required for its neuroprotective qualities? So this could be tested by deleting MGRN1's monoubiquitination domains and seeing if these MGRN1 mutants cause neurodegeneration as they did in the knockout models. Anyways, I hope everyone enjoyed this paper, and I encourage anyone with additional questions to post them below. I would love to discuss this paper. And I want to, again, thank the authors and the researchers for their amazing work. And thank you.